All right, welcome back. Today we're going to be looking at Module 7 within this video. And so, just as always, you can follow along the book. You can find this chapter starts on page, or module, starts on page 199. The PowerPoint can be found in D2L, the same place you found this video. Keep an ear out for the 4 random facts that we'll be giving you for the quiz for the lecture portion. And then also don't forget to do the quiz for the chapter itself. It's a longer module. So today, module 17, we're looking at vision and our ability to perceive and sense and all that kind of stuff. Kind of a follow-up, obviously, of what we've been looking at before, compared to like the next chapter, we'll be looking at all the rest of the senses. So vision is our strongest sense, or our dominant sense for an average person, assuming everything is working correctly. If you, for some reason, lose your sense of vision, or if you're born without a sense of vision, like your eyes don't work properly, the brain can actually rewire itself, and often has will, to utilize the space that would be set aside for vision for the other senses. So a person who's born blind, their other senses are generally significantly stronger than their sense of sight, obviously, right? But compared to average person, their senses are much stronger. But for an average person, the sense of sight is our go-to. So a large percentage of the brain is set aside for it. To go along with this lecture, I have several videos included. So if you look at this week's content information, you'll find 4 different videos actually. Bo Lodo, Optical Illusions, show how we see. Nathan S. Jacobs, how optical illusions trick your brain. Colm Keller, how we see color. And Brian Kett, what it's really like to be colorblind. These are worth taking a look at, especially the optical illusion ones, as far as kind of understanding how we perceive things versus how we see things, like as far as the sense of seeing, or the sense of sight. So take a look at those. The colorblind one is actually really also quite fascinating, kind of helping you understand what that is and how that works. But with that, without further ado, let's get moving through this module. So like I said, Module 17, Slide 2, light, energy, and eye structure. So how does the eye work? This is a super simplified version of how the eye works. So it'll give you the basics. If you really want to go in depth, take a bio class, basically. So, understanding light first. Wavelengths. Okay, so the distance from the peak of one light wave or sound. Okay, not necessarily just in light. Light and sound basically work in the similar way, just different forms of energy. The peak of the next. So you're going to measure the distances, and that'll affect the frequency, basically, right? How often that wave hits the given sense. Okay. Electromagnetic wavelengths vary from the short blips of cosmic rays to the long pulses of radio transmissions. Some of these waves can be miles long. Some of them are microscopic, basically. Okay. The intensity is about amount of energy in a light wave or sound wave, which influences what we perceive as brightness or loudness. The intensity is determined by the wave's amplitude, or the height of this. So if you're measuring a wavelength of light, the taller it is, the brighter the sensation would be of that experience. The hue is the dimension of color that is determined by the wavelength of light. So if you see green, it's because you're seeing a reflection of a certain wavelength of light. Excuse me. Woo! A little dust got in there. So a certain wavelength of light that allows you to see green. If you see blue, same thing. It's a different wavelength. There's a different distance between the two, so on and so forth. Okay, all the colors of the rainbow kind of a thing there. All right, so the stimulus light energy. So what is seen as light is only a thin slice of the broad spectrum of electromagnetic energy. If you really want to learn about this, you need to take a physics class. I actually looked at being a physicist for a short amount of time until I realized just how much math it was going to take. And I was like, no, don't want to do it that badly. But anyway, the portion visible to human extends from the shorter waves of blue-violet light to the longer waves of red light. So basically the spectrum of the rainbow, the Roy G. Biv, right? Red, orange, yellow, blue, green, red, orange, yellow, green, blue, indigo, violet. That spectrum is all of the different wavelengths that we are capable of seeing and perceiving. Okay. There are other organisms that can actually see different wavelengths. So like for instance, to give the example, bees, for instance, cannot see what we perceive as red, but they can see ultraviolet red or ultraviolet light, which we cannot perceive without special tools. It's actually how night vision equipment works. It'll pick up a different spectrum of light, and it actually emits light waves that are similar to that, like a flashlight, and then it gives you tools that allow you to pick up those waves. That's one version of night vision. The perceived hue in a light depends on its wavelengths, and its brightness depends on its intensity. So if you have a 1,000 lumen flashlight, the wavelengths would be very tall compared to a 100 lumen flashlight, for example, right? And compared to a candle, those would just be like ridiculously tall. Okay. So next slide, slide 4. If you look at this one is actually found on page 200 of our book as well. But it gives you it shows you the little tiny sliver of all of the potential energy light waves that are out there that we are actually capable of perceiving. So yeah, I mean, it's just that little teeny tiny rainbow there in the center is what we can see. When they're all combined together, we perceive it as white light. Okay, you break white light apart and you get a rainbow. So that's basically what you got. Okay, keep rolling. This slide is also found on page 200. Slide 5, physical properties of light waves. So it gives you just an example of like differences in wavelength. Higher frequency would equal bluish color. Longer wavelengths would equal reddish colors. And that's just the distance between the peaks of two different waves. And then the height of the wave would give you the brightness of a color. So like neon colors and things would be actually a more intense wavelength, which makes them seem more intense, right? This pencil, if it looks as bright as it does to me, is a pretty bright green. So this would have a relatively large amplitude or greater amplitude compared to, let's see, do I have anything that's kind of dull? Compared to this kind of yellow color, just dirty yellow color. Okay, so those are the differences in the spectrum there. Okay. And that's pretty much it. That's all I can think of to say about those. We'll keep on rolling. The fact that you can actually see those as different colors, though, it's going to be the eyes being able to detect those light waves bouncing off. And also, when you actually see a color, so for example, when you're seeing this green, the reason you see this as green, this is my son's, it has a little monkey all over it. The reason you see this as green is because this pencil specifically has green pigment in it, which allows it to absorb every other wavelength of light except for green. So in reality, this pencil is actually every color but green. It is reflecting green off, and that is what your eyes then pick up, is the wavelengths of light coming from this. It hits your eye, and you can see that wavelength as green. Okay. All right. That might be more than you wanted to know about that, but anyway, that's interesting to me at least. Slide 7, light energy and eye structures. Oops, I'm sorry. Slide 6, go back. Okay. The eye, I didn't talk about that yet. So you have different portions of the eye. You can see images of the eye on page 200 and on page 201. There's differing levels of kind of eye structure. We'll also have a slide coming up here on slide 8, I think it is, if I remember right. So yeah, the eye. The cornea is the portion of the eye through which light passes to the pupil and lens and is bent to help provide focus. Basically, it's going to intensify the quality of light coming through, essentially. Okay. The pupil is a small adjustable opening through which the light then passes. It's literally just a little hole in your eye, but it can contract and extend given the iris and the like. Okay, so a colored muscle. The iris is the colored muscle surrounding the pupil that controls its size. So when you have a lot of light, let me turn this toward me. Okay, the pupil gets quite small. When you take the light away, let me do this. If you can still see my eyes, take the light away a little bit more. The pupil gets bigger. This is weird. Anyway, okay. And you can see them flex and stuff. You can do this in the mirror. If you go into the bathroom when it's dark and have a flashlight with you, you can turn the flashlight on and hit your eyes, and you can see your eyes go from really big to really small really quickly. But anyway, there's a good chance you did it as a kid if you haven't done it recently. Yeah, it just controls how much light is coming in through the opening of the pupil. The lens is going to focus the incoming light rays onto an image on the retina on the eyeball's sensitive inner surface. If you've ever actually got to dissect an eyeball from an animal or something like that, the lens itself is actually legitimately like what you would expect, like a camera lens or something like that. It's kind of a plasticky feeling. It's firm and it can be flexed to focus. And so that's what allows basically that it can get that clear image. It does it amazingly quickly. This is what allows you to see from everything from just like a couple inches away all the way to the mountains way off in the distance, right? 20, 30, 40, 50 miles. Technically even to the stars, light years away. The eye is able to adjust distances very rapidly while you're young, assuming your eye is working properly. Okay. The older you get, it starts to deteriorate. It takes longer and longer for the eye to actually focus at those different distances, which you might notice if you are getting older, like myself, moving from like 2 inches away to the distance. It's a noticeable moment where it's unfocused and then it moves into focus now. When I was younger, I didn't have that at all. So things change. After entering the eye and being focused by the lens, light energy particles strike the eye's inner surface, which is the retina. Okay. Next slide, slide 7. Light energy and eye structure, part 2. The retina. The retina contains 2 types of receptors, rods and cones. Okay. Has layers of neurons that begin the processing of visual information. So these are little points that are going to absorb the energy from the light and turn that into a signal that the brain can then basically interpret as sight, right? You see it as color. You perceive it as color and shape and motion and all these kinds of things. Accommodation is the process by which the eye's lens changes shape to focus near or far objects on the retina. Okay, so it'd be kind of like if you have a magnifying glass. I was thinking, what am I doing? If you have a magnifying glass, if you've ever tried to like focus it in where you can burn things, or start a fire when you're camping and things like that, or if you've ever tried to, you can technically do it where you can focus it and you can get an upside down image of what's like if the light's coming in from this side over here. And you put your lens here, you can actually create an upside down image on like a wall or something over here if you can focus it in. Our lens is basically doing exactly the same thing. It's trying to get it to the proper focus, like a camera kind of, for the given distance that you're trying to focus upon. Okay. Slide 8. This is just an image of kind of how the eye is working. So you have the iris, cornea, pupil, and lens all working there in the surface. Focusing the light. And if you notice the candle light in the back of the eye, again, you can also see this image on page 200. There's a candle in the front, and it's flipped upside down. The lens, just like a magnifying glass, would actually, because it's curved, flips the image that you're perceiving or that you are receiving upside down. And it basically creates an imprint on the retina. The amount of light coming in will cause a reaction in the retina, a chemical reaction, which then is translated into neural impulses, which is then sent on to the brain to be utilized, to be processed, and then to be like kind of broken down so you can understand what it is that you're actually looking at. Okay. Every eye has a blind spot, right where the optic nerve comes in. There are no cones or rods there, so you actually have a tiny, tiny spot in the back of the eye where you can't perceive anything. And in fact, we have a blind spot in our eye. It is almost impossible to see, though, because of the fact that I think I talked about this in the last one, that our eyes are constantly moving. That constant motion essentially eliminates the effects of the blind spot for the most part. Okay. So it generally is not perceived, even though it is there. All right. It's also, the blind spot is slightly offset from the center, and so it's generally slightly lower. And so therefore, what you are directly looking at, you can actually still perceive. It's just right below what you're directly looking at that you lose in the process. Okay, slide 9. The eye to brain pathway. Light energy particles trigger chemical reactions in receptor cells, rods, and cones that form an outer layer of cells of the retina at the back of the eye. Rods are the retinal receptor, and rods and cones are going to basically be every critter, well, almost every critter. There's a couple that have a very different eye structure. Scallops are one that has a very interesting eye structure. But anyway, as far as I know, all mammals are going to have rods and cones, but they're going to have different levels of them, different layers of them, and different sets of them, which allow them to see different levels of lighting and things like that comparatively. Okay. Rods are the retinal receptors that detect black, white, and gray. They're sensitive to movement. So this is actually the part of the eye that we have the majority of them on the external portion of the retina inside there. And essentially, this is where we get our peripheral vision. Okay, they're not real good at detail, but they're real good at catching motion and movement. And it's what allows us to see in relative darkness. Okay, twilight vision. When the sun has just set and you get that weird stage where everything is kind of weird feeling. It's like there's like an hour before the sun comes up and an hour before the sun goes down. You have that awkward stage of lighting. It's actually because your brain is trying to figure out if it should be engaging the cones or just the rods. Once it sets and it's dark and maybe you just have starlight or moonlight, it knows the rods are what get engaged in that environment. Okay. Which is why you can see movement generally, but you're gonna have a harder time seeing like details in the dark. Cones are receptors concentrated near the center of the retina, and they function in daylight or well-lit conditions. And their whole thing is to detect fine detail and color. That is their primary. That is the fact that you can see colors is because we have these three different cones that basically will get triggered from different wavelengths of lighting. Okay. So yeah, we have more rods than cones also. So slide 10, the retina's reaction to light, and this just kind of gives you the process. You can find this also on page 201. I'd actually recommend looking at the book in this case because it gives you a little bit more details around it. So just take a look at that. But it shows you kind of the processes by which the information is transformed into neurotransmissions. Slide 11. Rods and cones. Cones are sensitive to detail and color. Rods are sensitive to faint light, which we already said. We have significantly more rods than cones in our eye. So the average person will have 6 million cones and 120 million rods. In a lot of ways, the rods are actually more useful, at least as far as like because of darkness and things like that, right? But the cones give you that ability to actually do detailed work and to see what it is that you are doing. Location in the eye, the cones are going to be pretty much all in the center. The rods, there are some in the center, but they're primarily going to be around the periphery of that center point, right? That peripheral side of things. Sensitivity in dim light, cones are low. In fact, cones to some extent basically don't engage at all if the light gets too low. Rods are at their peak when they're in the lower lighting. Color sensitivity, rods basically don't have any. You see in black and white. Cones are high. That's right. You can see color. Detail sensitivity, rods are very low, which is why you don't have real clear detailed information in your, like, right, if you put your hand up in your peripheral vision, I can see that's a hand. I can see that there's 5 objects there, 5 fingers wiggling at me. But I really don't have a very good idea of the coloring. I can't see the lines in my hand and all that kind of stuff, right? It's picking it up. The hand is there. It can really see the movement, but I can't actually see the details. When I look at my hand directly, I can be like, oh yeah, there's teeny tiny little cracks, itty bitty little lines in your hand, things like that. Okay. And that's the difference between the rods and the cones. Okay. Slide 12, information processing in the eye and brain, retinal processing. So the optic nerve carries the neural impulse. So the eye is basically, the rods and cones are triggered. They get insulted. No, they fire off basically and sends an electrical impulse into the optic nerve, which is a very large nerve coming out of the back lower portion of the eyeball, through the brain, crisscrosses in the middle, and ends up here in the back. Okay. These neural impulses are carried from the eye to the brain. The blind spot is the point at which the optic nerve leaves the eye where no receptor cells are located, right? The fovea is the central focal point in the retina around which the eye's cones cluster. So again, if you look at that image of the eye, you'll kind of see that. But if you look at this next image, you can find this on page 203 if you're following in the book, or on slide 13 in our lecture. The pathway from the eyes, they go from, again, both eyes are going to go to both sides of the brain. So the left eye isn't just to the right side of the brain, and the right eye isn't just the left side of the brain. But there's a crisscross where some of the information goes one way and some of the information goes the other. Both eyes are going both ways. Okay. The visual cortex is in the very back of the brain, basically right there touching the back of the skull, essentially. Okay. So yeah, it passes through the thalamus as it goes, triggering different things in that process. Okay, first random fact. The Mona Lisa was stolen from the Louvre in 1911. And interestingly, more people came to actually see the empty space where the Mona Lisa used to be than they came to actually see the Mona Lisa. So the museum actually had more traffic after it was stolen than when they had it, which is kind of fascinating. But yeah, the Mona Lisa was stolen in 1911. Okay, next slide, 14, color processing. So color processing is a two-stage process. Young-Helmholtz trichromatic theory is the retina's red, green, and blue cones respond in varying degrees to different color stimuli. Essentially, certain wavelengths are going to trigger certain cones, right? We have 3 cones. We have ones that read blue, we have ones that read red, yellow, and we have ones that read green. Okay. When you have those 3 combined, you see white, when they're all triggered at approximately the same rate. But otherwise, essentially, if you're looking at something and it picks up the green ones, it sees it as green because those rods or those cones are more stimulated than the ones that would pick up the blue and the red. Okay. Haring's opponent process theory, cones responses are then processed by opponent process cells. In other words, they go through and that information basically gets broken down and processed there in order to get sent forward. So, feature detection, slide 15. Feature detectors are specialized nerve cells in the brain that respond to specific features of the stimulus, such as shape, angle, or movement. There are different parts of the brain that basically are set aside for these different aspects of sight. So if you see my hand moving on the screen, right, there's a part of your brain that's registering the color. There's a part of the brain that's registering the shape of my hand. There's a part of the brain that's registering the fact that it's moving back and forth. Okay. And right, even a bigger part of the brain that's like detecting how close it is, apparently is to you and how far away. Okay. All of those things are broken down into the different parts of the brain and then recombined to give you your perception of what it is that you are seeing. So these cells receive information from the ganglion cells in the retina, and they pass that information to other cortical areas where teams of cells, supercell clusters, these are kind of like the big cities of within your brain, respond to more complex patterns. Okay. Next slide, 16, face recognition process. So the ability to recognize faces is basically located right down here below the ear. In social animals, this portion of the brain is relatively large because it's an important aspect, right? Humans, we have a large percentage of our brain, relatively, that is basically set aside to recognize faces and to recognize individual faces. This is also why we see faces everywhere, right? You look at the clouds and you're like, oh, that looks like a big face staring at me. You're like in the rocks and the mountains and things like that, in a weird gnarly tree. Our brain is like constantly looking for faces, and because of that, we see them everywhere. It's in the large right temporal lobe area, approximately. Okay, and if you see this image, it is in. You see this image on page 205 that we're looking at on the PowerPoint here, slide 16. But yeah, its whole point is basically to recognize faces. Okay, if this part of the brain gets damaged, you basically lose the ability to recognize a face. You might be able to tell it like, well, that is a face because I can put it in proportion to like there's their head and it's connected to the body, and so therefore that's the face. But the ability to then determine whose face that is gone. Okay, so I couldn't tell the difference between a random woman on the street, my wife, and my daughter. Looking at just their faces, if this part of my brain was damaged, I wouldn't be able to tell which one was which. Okay, if I talk to them and stuff, then all those other cues would give me who they are. But initially, you couldn't tell. Okay. Random fact number 2. Okay. somebody hid an episode of South Park, a full episode of South Park, inside the game, the video game Tiger Woods 99, as an Easter egg. It was one of those you could like stumble upon it. When EA Sports found out about it, though, they had to basically recall all the games. It was a massive recall. But yeah, there was a South Park episode hidden inside Tiger Woods 99 as an Easter egg find originally. So if you have one of the original versions, you might still be able to find it. Okay. Slide 17, parallel processing part 1. Parallel processing is the brain's ability to do many things at once. We talked about this a little bit in one of the first modules. But this is the fact that you have your conscious experience, which is actually a relatively small percentage of what your brain is doing. You have all these different things happening at the same time where it's accessing memories, it's accessing what all of your senses are bringing in and kind of ciphering through it to figure out what is worth pushing forward into the conscious thought. It's giving you interpretations of what it thinks that it is that you are seeing. All of that is happening at the same time. Okay. And then getting pushed forward. And this is that parallel processing standard. So a visual scene is first divided into subdimensions. So if I'm looking at something, it's going to break it apart. You can see an example of this on page 206. It breaks it apart like outline. It breaks it apart by movement. It breaks it apart by textures and colors. All these things are broken apart into these subdimensions and then brought back together in our perception. So perceptions are constructed by integrating separate but parallel subdimensions of the various stimuli. Okay, slide 18, we have the same image that we have on page 206. Okay. So you have motion, form, depth, color, broken apart, processed by different parts of the brain, and then brought back together. The interesting thing is that we know that our brain does this. We know for a fact this is what our brain does. We don't know how, and we don't know why. Okay. At this point, if you could crack the code and actually figure out how the brain pulls it apart and then puts it back together, you would be a world famous psychologist / biologist and probably be remembered in all the science history books and everything for the rest of eternity, or at least for as long as our culture lasts. But yeah, they're still trying to figure this one out. Because it's potentially one of those, if you can figure it out, you might be able to solve a lot of other problems that we experience in vision and different things like that. So, interesting. 19, perceptual organization. Gestalt is an organized whole. There's a Gestalt theory of therapy, but this isn't exactly what that's talking about. Gestalt psychologists propose principles used to organize sensations into meaningful wholes. That's actually where the therapy kind of, to some extent, comes through also. It's looking at how we construct our views and how we're constantly trying to figure out, how to finish and complete things. Okay, so in perception, the whole may exceed the sum of its parts. You might have something that is more than what it apparently is, right? If I show you a flag, like an American flag, nope, don't got one. If I show you an American flag, that flag is more than just red, white, and blue colors, right, on a piece of cloth. It represents something much more in its whole of what it is. It represents an entire country. Okay. So that's a perception. It's above and beyond what the senses can tell us. Okay. We filter incoming information and construct our perceptions then from that, like we talked in the last module. We bring in all that information, we process it, and then we figure out what to do with it, right? So if I show you a pin, I have lots of pins, I'm realizing. If I show you a pin, the fact that you recognize this to be a pin that's not immediately evident. If you just took it as what you are actually seeing, all that you're actually seeing is a blue stick with a weird little nub here on the top, right, and some little joint points. But your perception then takes experiences from the past, combined with what your senses are taking in the present, and gives you the whole picture of what it truly is, or what it apparently is, at least. Questions. This is where I'd actually recommend, make sure that you watch those videos that I suggested earlier. So the Bo Lodo's Optical Illusions and Nathan S. Jacob, How Optical Illusions Trick Your Brain. Take a peek at those, at least one of them. Okay, they kind of go over similar information, but it really does give us a lot of details on how we perceive things, how we take in information and process it to understand how the world works. But anyway, Form perception part 1, slide 20. How do we organize and interpret the shapes and colors into meaningful perceptions? People tend to organize pieces of information into an organized whole or gestalt. Okay. You can see the image that we're looking at on this slide on page 20, on slide 20 or on page 207. But when you're looking at this, there's a little X in the middle of all those blue splotches. Okay, stare at the X for a second. What do you see? There's a good chance you saw it even before you looked at the X, but what do you see? Okay. Do you see circles with a cube in front of them? Do you see random blue splotches with just like white breakup points in between them? Right. Your perception of it. What most people will see is, in fact, a blue cube. Either a blue cube with blue spots behind it, white cube with blue spots behind it, or a well, that's generally what you're going to see. And depending on which way it's angled and things like that, it can change. Our brains don't like things to be broken up, right? We don't see a bunch of weird little random blue spots generally. Our brain sees that there are lines that are aligned with the lines below it, and therefore they must be connected, right? If I go like this, people assume that these align with each other and that I'm representing something, like I'm holding something. I'm actually just making two like crab claws, right? But if I go like this and I'm like, people assume that I've got a sandwich in my hand, right? We fill in the gaps. We naturally want to fill in the gaps. That's a gestalt. That's the brain's tendency to gestalt things. Okay. Slide 21, and you can see the same image on page 207 down at the bottom. Form perception part 2. So figure ground, the organization of the visual field into objects or figures that stand out from their surroundings, the ground. In this image, what do you see first? Do you see a vase or a cup? Or do you see two faces staring at each other? Okay. It really depends on, is it two black faces that are in black with a white background, or is it a white cup on a black background? Okay. And depending on which way, and oftentimes you can just kind of flip back and forth between the two when you look at it, once you can recognize the two things. But it's reversible, right? Our perception is really all that matters there. Neither answer is correct or incorrect. They're both correct and they're both incorrect at the same time. Okay. 22, form perception part 3, grouping, the perceptual tendency to organize stimuli into coherent groups. Just like that square, that cube, right? We like things to be connected and to be completed. We don't like things to be broken apart. So proximity, grouping nearby figures together, is a natural tendency. Okay, continuity, perceiving smooth, continuous patterns rather than discontinuous ones. Closure is the filling in the gaps to create a complete whole object. If you go to the next slide, or if you turn the page to page 208, you'll see images. On page 208 is at the top, but looking at grouping. Okay, so slide 23. Human minds use these grouping strategies to see patterns and objects. So A is an example of proximity. Basically, there's a tendency for people to see 3 columns rather than 6 separate lines, okay, because they're a little bit closer together, so therefore they must be connected. In image B, we have a wiggly line over a straight line, but we generally don't see it as a bunch of half circles and line segments. We see it as a continuous line with a squiggle line going back and forth across it. Okay. Image C, the image on the left, you'll see looks very much like just like a floating triangle. It's kind of like what we saw on slide 20 with the cube, right? It's like a floating triangle in front of circles. When you close the circles off, you can still see where the triangle could be, but because they're closed like little C's, our mind is more comfortable with them each being 3 separate circles, or 3 separate C's. They're next to each other. Okay, but when they're open, our mind's like, oh, there must be like a floating white triangle in front of it, typically. Okay. 24, depth perception. Depth perception is the ability to see objects in 3 dimensions, although the images that strike the retina are 2-dimensional. It allows us to judge distance is present, at least in part, at birth in humans and other animals. It's barely present in humans. Okay, when we're born, interestingly enough, even though our sight becomes our most dominant sense, it is the least developed at birth. Um, you can see from approximately 4 inches to 30 inches at birth, which is important, right? Um, at that age, you really only need that much because all you need to be able to see is see a face from when someone's holding you, okay, and find a nipple. Those two things are essentially all you have to be able to see. When we're born, our eyes also aren't in sync with each other. And so, if you ever looked at a newborn baby, there's a tendency they might be looking at you, and then all of a sudden their eyes are like, okay, it's because they're not synced up. We don't have binocular vision until around 2 months to 4 months, where they actually kind of come in line, and at that point we are then capable of perceiving depth, but we don't have very much experience with it, and so therefore our perception of depth at that point is generally, can I reach it, or is it out of reach? That's about as far. So I know it's like close enough for me to reach, and or it's out of my reach, and that's my extent of my perception of it. With motion, once we get mobile, start crawling, we very quickly develop a stronger sense of depth, and the ability to tell like how far away something is, how long will it take me, should I like go off this edge, and if so, how far will I fall? All of those kinds of things. Okay, that happens usually within a week or two of actually becoming more mobile. The brain quickly wires itself to keep you relatively safe in making your judgments. Slide 25 actually shows that. It's also found on page 209. The visual cliff. Tests of early 3D perception. They did these experiments where basically they took a bunch of babies who were just starting to get mobile, and they put them on a table that had little checkers and an obvious drop and checkers. The checkers stayed the same size all the way through, so there's an obvious difference in size, which gives you the perception of depth. And then they put a piece of clean glass over the drop. So it was a visual cliff with no actual danger. Okay. What they found was initially, if they had just within the day or two started moving, they would fairly confidently go right on out onto the glass. Okay. After just a week, it was very difficult to encourage them to go out onto the glass. And they've actually found this even with other animals. They've tried it with like goats and pigs and different things too. All animals are afraid to step out onto the glass because of the perception of the fall. Okay. So crawling, no matter when it begins, seems to increase the infant's fear of heights, or at least caution of heights. Okay. Slide 26, depth perception part 1, binocular cues. 2 eyes help with perception of depth, right? If you cover an eye, you lose the majority of your depth perception. I can have a rough guess about how far things are away by expected size and things like that, but I lose my ability to really determine things well. Binocular vision essentially is when your eyes are synced up together. You can tell about how far away something is because your brain does a really amazingly fast amount of math, and it figures out the angles of your eyes in comparison to each other, and it calculates how far the thing must be given the depth or the change of your eyes. It's extremely effective. We have extremely accurate perception up to about 10 or 12 feet. Beyond that, it gets harder. You can develop it with practice, but the binocular cues are less useful beyond 10 or 12 feet. Okay. That's basically what retinal disparity is. Binocular cue for perceiving depth. By comparing images from the retinas in the 2 eyes, the brain calculates distance. So again, the angle at which your eyes have to be in order for them to come to a line allows you to then perceive depth. It's what basically 3D filmmakers are forcing upon you, which is why sometimes 3D films can actually give a lot of people headaches. Is it's forcing something upon you that your eyes are having a difficult time doing on their own, because there really isn't that much. The distance isn't actually changing; it's the perception of the distance changing. So they force it with the glasses and everything to make it to make your eyes see the 3D happening. Okay. So yeah, that's going to be kind of what it works. So beyond 10 to 12 feet, our eyes don't do this as well. Again, with practice, like I had a friend. He was a Navy SEAL sniper, or at least a sharpshooter. But anyway, he proved it to me a couple times with his shooting is amazing. But he is amazingly accurate. If you sit outside with a rangefinder, you can say how far away is such and such a tree, and he could get within about 5 feet of it, even out to well past a quarter mile. I've never seen anybody be able to do it like he does, as accurately as he does it. But that's with practice. He had training. He also had lots of tours that he actually put it into like life and death practice. And so that was an important aspect of keeping himself alive, right? But that's something that it doesn't necessarily come as easily if you're not given that kind of training. Okay, slide 27. Depth perception part 2, monocular cues. Depth cues such as interposition or linear perspective available to either eye alone. So like things like relative height, relative size, interposition, linear perspective, light and shadow, relative motion. All of those things are visible with just one eye. Okay. Motion perception is the stroboscopic movements and 5 phenomena. These are going to be all those things that basically your eye, again, you don't necessarily need 2 eyes in order to perceive these things. Excuse me. Okay, let me see. Yeah. Trying to think if there's anything else I need to say about that. I think that's pretty much it. Yeah, we'll keep on rolling. 28. Perceptual constancy, part 1. Perceptual constancy, objects are perceived as unchanging, having consistent color, brightness, shape, and size, even as illumination and retinal images change. If you watch those videos on the optical illusions, you'll see this in practice, and that's why one of the reasons why I highly recommend watching those. Okay, random fact number 3, and I'll come back to this. Batman and Predator are in the same fictional universe and have been together 3 times in comic books. So yeah, there you go. I think they first appeared together in the mid-90s, if I remember the facts right, but there you go. So yeah, if you can see an image, so you can see an example of this. Sorry, I'm coming back to perceptual constancy part 1, slide 28. You can see an example of this on page 211. Down at the bottom, there's a cube with 3 apparent differences in light. The blue spots on those cubes are exactly the same color, but we perceive them as different because our brain basically is reading the light cues around them and saying, well, they can't be the same color because the light cues say that it's different. Okay, watch those optical illusion videos and you'll see this in action. Slide 29, perceptual constancy part 2, color and brightness constancies. So color constancy, perceiving familiar objects as having consistent color, even if changing illumination alters the wavelengths reflected by the object. So there you go. There's what that one is. Brightness constancy is similar, depends on context, right? It's going to be showing us all of like what we do expect. On page 212, there's actually an image, and you can see image A and image B are actually the same color. Okay, in that one, even though they look different because our brain is like it's under shadow or under light. Slide 30, shape and size constancy. So shape constancy, perceiving the forms of familiar objects as constant even when our retinas receive changing images of them, right? And it gives you the example of the door being opened. You can see this at the bottom of page 212 or here on slide 30. What we're actually seeing is a rectangle with rectangles inside it. And then, what is it, a trapezoid, where the rectangle suddenly changes angles, right? And then a more intense version of that. But we see it as a door opening rather than different shapes, because we assume, given our experience of doors, we assume that that door is actually remaining the same. So therefore, as it comes closer, right, like if it's my hand, as it comes closer, my hand, we assume, is actually staying the same shape. It's not that I'm changing the complete form of my hand. It's just that one part of my hand is getting closer to you and then further from you. Okay. That perception is utilized in art and things like that also. So that we don't see it as a changing shape. We see it as a rectangle at a different angle. Okay. Slide 31, and last random fact before we close it. Amber-colored rear turn signals have been statistically proven to reduce collisions by about 28%. Use your blinkers, in other words. Okay, that's my goodwill for the day. 31, experience and visual perception. So restored vision and sensory restriction. The effects of sensory restriction on infant cats, monkeys, and humans suggest there is a critical period for normal sensory and perceptual development. We develop these senses again in those early months of life as humans, but cats and things also are going to be doing the same thing. There's a point where if they don't get it, they're going to struggle to develop that perception later. So without stimulation, normal connections do not develop within the brain. And so without those connections, perception is much more difficult. Perceptual adaptation is the ability to adjust to an artificially displaced or even inverted visual field. You can put glasses on that shift your perspective. Okay, and in some cases, actually even just flip-flop you completely. And it can be very disorienting, especially at first. But over time, your brain can actually adjust to it if you've had experience with vision. Usually about a week in, if you wire them basically constantly, your brain will adapt and be able to basically function well, even upside down. I saw a video, maybe I'll see if I can track it down. But I saw a video where a guy actually created a third-person module where he wore this chest rig and had a branch or a big pole coming out the back with a camera mounted on it, and then he had like virtual reality goggles on, and so he could only see through the perspective of the camera behind him and above him slightly. And it took him, at first he actually had a lot of problems with motion sickness and stuff, but eventually he got to the point where he could function pretty well, only being able to see from behind him in a third-person perspective. I'll see if I can track that down. If not, just you can like Google it or YouTube it. I'm sure you can probably track it down. But so yeah, nature and nurture. This is a big part, right? Nature is going to be what we are born with at this point. The nurture portion, like the experiences that we have, are going to begin in this early stage of life, are going to begin to shape our perceptions that we will then take into basically the rest of our lives. And so we are reliant upon experience, utilizing our senses in order for them to be fully developed and properly developed later on. Okay, giving you all 4 facts, giving you basically all the information from this this module. It's a big one, right? Make sure you read ahead. In the next video, we'll be looking at all of the rest of the senses all at once. There's a little bit of repeat. Some of the stuff, again, sound and sight kind of use the same terminology as far as like wavelengths and all that kind of stuff. But so we were prepared for that. Make sure you do the quizzes, right? The quiz that is connected to the 4 random facts, as well as the chapter quiz. Beyond that, I think you should be good to go. I will see you all in the next video when we look at the next module, looking at all the other senses. Have a great day, everyone, and I will see you in the next one.